Bismillahirrahmanirrahim. This is the second video on chapter 7, which is transport in plants, and this is the new syllabus, which is 2023 syllabus. Now, the points left in the previous video is one is this one, 7.13, and 7.23, 4, and 6. So that is what we're going to handle in this video. Now, 7.1 syllabus point 3, investigate using a suitable stain the pathway of water in a cut stem, not in a full proper plant, not in a potted plant. It didn't say in a potted plant. Because a potted plant, must remember, a potted plant will have roots in it. Here we are not saying that. We are saying a cut stem. Please be very careful when you are reading. Some of you read, yet you do not read. So it is a cut stem, and we're going to put this. So let's take a beaker. So we take a beaker and we put some water in it. So here is the beaker, and we've added some water to it. And now we add a dye to it. And this is a red dye. Now the red dye is going to enter. Please remember, dye does not enter by osmosis. It will just move into the either we don't know xylem or the phloem. We are a little confused about that. And then it will go and enter, and then it will enter the leaves. Right? Now, if preferably this should have white flowers. Why should it have white flowers? Is because the white flowers would help us to decide, because then we can see the dye in the white flowers. So we can see the dye inside the white flowers, and the white flowers will turn red because the dye will have reached the white flowers. So preferably we should have white flowers, stem with white flowers. Now the question is that the biologists are in disagreement that does the water and the dye travel in the xylem or the phloem. So the question is, how will you investigate the pathway of water in a cut stem? So what you need to do is, now that you've left this, so you put the dye and you leave this for 6 to 8 hours, and you leave this in bright light. Why do you leave it in bright light? I wish there was a proper class and I would have asked you these questions, and then the sort of changad answers you all would have given me would have really made me wonder whether I was teaching biology or something else. And when we leave it in bright light, the stomata open and the water vapors diffuse out, so the suction force will be created, and this water will be sucked up in the stem. So bright light means stomata open, water vapors diffusing out. Water vapors, not water. Water vapors diffusing out, causing a suction force, with which we have talked about in the previous video. And the water is forced up the stem and enters the leaves and the flower. Now what we need to do is, we need to take a section. We need to cut the stem here, a very fine section of the stem. And then we need to take a slide. And then we need to put this section on the slide, and we need to examine it. Now, if you remember, in the previous video we talked about it. The stem, the vascular bundles are on the periphery. They're on the side. They're, periphery. Periphery means on the edge. Shayad Afridi caught the catch at the periphery. So what we have to understand is, now where do we see the dye? If we see the dye on the inside, then it is in the xylem. But if we had seen the dye on the outside, then it would have been the phloem. But of course, as you can see, the dye will be found on the inside. So that means the dye with the water has entered the xylem. So the dye is on the inside. So this proves the fact that water and the dye is transported in the xylem and not in the phloem. So the disagreement between the biologists is solved, because what we did is, we scientifically proved it. We made a slide of the stem. First we took the stem, the cut stem, we placed it in the dye and the water, then we left it in for 6 to 8 hours in bright light, and then we cut a section of the stem, a very fine section, a thin section, not a fine but a thin section of the stem, and then we placed it on the slide, and then we found where was the dye, where was the red color. Whatever the color of the dye, I'm not saying it's always going to be red, could be blue color as well. So this is how we would do this investigation, this practical. Now, the next topic that we need to study is the transpiration rate, the rate of transpiration or the transpiration rate. Now, how would we measure this? Now, measure this is somehow it is very difficult to measure this because we don't know how much water is entering the plant and how much water is escaping from the leaves. And the water is escaping as water vapors. So how do we measure that? Now, that's another difficult thing. So what we need is we need some sort of an apparatus, and that apparatus is called a potometer. And there are many types of potometers. One is a weight potometer, in which you just weigh the plant and then you wait after some time and then you see the loss of water is going to tell you how much there is loss in weight. And there are other types of potometers. Now here is a diagram to show you a potometer, and a potometer is a device used to estimate the rate of transpiration. Experimental method is the distance moved by an air bubble. So there is an air bubble and the distance it will move can be recorded every minute and used to indicate the rate of water uptake by the plant. You see, the volume can be calculated by using the formula pi r square h. But the length of the column, we can find out, okay, if the bubble was here and now it is here, so that means it has moved this distance. This distance is h, and if you know the radius pi r square h, we can calculate the volume. We can also calculate the volume because if it is moved here, now we open the reservoir. There's a tap here. You open the reservoir and then you let water flow in, and then this bubble is going to be moved back to the beginning or to the beginning here. So the volume of water which has been used up, you can calculate and you can say, okay, 2 ml or 3 ml, what was the volume of water that was transpired. But you see, now here's another very important thing. Now this is for the A star students to understand that what we are thinking is that this water has gone in here, 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 and out of the plant. This is what our assumption is. What is our assumption is that water uptake is equal to water transpired. Now this is slightly wrong. Why is it wrong? Because some of the water may have been used for photosynthesis in the leaf. Some of the water may have been used for turgor pressure. But what we are assuming, this is for only some of the students to understand this concept, that this potometer is only measuring water uptake because it is telling you, okay, the bubble moved from here to here. So that means so much water. Please understand this. I'm going to explain it again to you. We are going to assume that the bubble moved from, say, here, it moved here up to this place, it moved up to here. So that means this column of water, what has happened to this column of water? This column of water has gone in here and out of the leaf. But then the water might have been used. It's just like if you drink 2 glasses of water, well, you're not going to urinate 2 glasses. The water in your body is used, and maybe some of it becomes urine. So water, what we are assuming here is that the water uptake is equal to water transpired. It's practically correct because only 1% of the water is used up by the plant for either turgidity or for turgor pressure or for photosynthesis. So this is our assumption, that we are assuming that the water uptake is equal to the water transpired. That's why I gave you this. This is of course for the student who is going to be asked that one suggest question, that why is it wrong to say this is measuring the rate of transpiration, because it's only measuring the rate of water uptake. So this is one potometer. Then we'll show you another potometer. This is another potometer like you see here, the rate of water uptake can be measured using a potometer. So the leafy shoot is placed in the apparatus under water so there's no air spaces, and this whole thing is sealed here with Vaseline, so there's no water loss from there. The rate at which this bubble moves can be recorded by measuring the distance on the graduated capillary tube and the time using a stop clock. Number 3, water from the reservoir can be let into the apparatus to push the bubble back to the starting position by opening this tap. So when the bubble has moved here, Now we need to go back, so we need to set up the apparatus again. We must get about 5 readings. In science, we always get 5 readings so that then we can calculate the mean, so that we can remove the anomalous readings or the error readings. There might be an error. One of the readings might not go with a trend. Say it moves 10 millimeter, and then it moves 9 millimeter, and then it moves 20 millimeters, and then it moves 8 millimeter. Well, you know there's something wrong. This reading is probably wrong because the others is 10, 9, 8, and maybe another 10 here. So unless you do 5 readings, only then you can find out which one is the error. If you had only done one reading and it was 20, then you would have got this wrong. But you did 5 readings. One was 10, one was 9, one was 20, one was 8, one was 10. So one did not fit the pattern, the normal pattern. Another diagram of a potometer, and you can see here, you can pause the video here, have a look at this, and understand how and what we are measuring, and how this is another type of potometer. But please be very comfortable with all different types. Basically, the core concept is the same, that the water has to move up into the plant and out of the plant, and there is an air bubble somewhere, and that movement of the air bubble helps us to find out the rate of transpiration. Another diagram showing you a potometer. Please pause the video here, read through it. Transpiration can be measured using a potometer. Cut plant stem is sealed into the potometer using a rubber bung. Air bubble is introduced. The distance the bubble travels shows how much the stem has taken up. This gives an indirect measurement of the rate of transpiration. That is, it measures water uptake. Now, what we need to find out is what are the factors that affect the rate of transpiration. Now, I've made a mnemonic for it, HALT. Basically, it's humidity, air movement, which is also wind speed, which is in your syllabus, the word wind speed has been used, light and temperature. And light is light intensity. So we've got to study these 4 factors on the rate of transpiration. So naturally, we're going to use a potometer. Now, the first thing is that if we have to study, let's look at the first one is temperature. If we have to study the effect of temperature, well, all we have to do is keep these in different temperature water baths. So the first water bath maybe could be at 30 degrees Celsius, the next could be at 40 degrees Celsius, and the next could be at 50 degrees Celsius. And you should take 5 temperatures, and then you should keep them in a different water bath. And you should just measure the rate of transpiration. Now, naturally, what is going to happen? As you increase the temperature, the rate of transpiration will increase. Why? More the temperature means more kinetic energy. More kinetic energy. So more what? More diffusion of water. Here we are not talking of osmosis because water is moving in a column. More diffusion of water, more water moving up the column, and more evaporation. Because you see, evaporation is the basic need before transpiration can occur. The liquid water has to convert into a gas, so it has to be evaporation. So more kinetic energy will mean more diffusion, will means more evaporation. And water vapors also have to diffuse out of the leaf through the stomata. So water vapors diffusing out of the leaf through the stomata. So the effect of your syllabus says you've got to investigate, number 2, and then you have to explain why increasing temperature would increase the rate of transpiration. Now the second factor that we need to investigate is wind speed. So for that, of course, we will need a fan. I know you might not approve of this fan of mine because it's facing you rather than facing the potometer. So a fan, and you would measure the movement of the air bubble using a fan. You can, of course, change the distance of the fan. You can maybe make it move a little away, and you can move it away from it, or you can move it nearer to it. And you can do this experiment and see is the movement of the how is the movement of the bubble. Is it moving faster? Is it moving slower? Naturally, I mean scientifically, more the wind speed, if the fan is nearer to the water meter, more rate of transpiration. Why will it be more rate of transpiration? Any answers? No, I'm not getting any answers. Everybody is fast asleep. So what will happen is that when the air, there is air movement, so the water vapors which have diffused out of the leaf will be carried away. They will be carried away. So this will create a zero gradient outside the leaf. So more water vapors will diffuse out of the leaf. This is a leaf and the water vapors are on the outside. But when there's a fan blowing, it's going to carry away the water vapors. This is the thing, and this is the water vapors. So when there is going to be air movement, the water vapors are going to be carried away and they're going to be moved away from the leaf. So then there is zero water vapors outside and 100 water vapors inside the leaf. So the gradient is maintained and water vapors will diffuse out, or maybe even if they are 10, but 100 to 10, so water vapors will diffuse out. So more the rate of transpiration. It's like if you want to dry your hair, so you just sit under the fan. Or of course you use a hair dryer, then of course it's warm air. Then of course will result in the water evaporating faster from your hair if you want to dry your hair quickly. So more the wind speed, more the rate of transpiration. And all you need is a potometer and a fan to prove this. So you'll do an experiment with a fan and then you'll do an experiment without a fan and see how the difference is. Or you can increase the speed of the fan and study it, more the wind speed, more the rate of transpiration. Now the third thing that we have to investigate is light intensity. So light intensity means that what we need to do is we need to then of course either change the distance of the lamp, move it away from it or move it closer to it. So we can move the lamp, either we can do that or we can have a dimmer switch in it. If you have a dimmer switch in it, of course then it is easier for you to change the light intensity, or you can change the number of lamps. Now, you have to understand, why would increasing light intensity increase the rate of transpiration? Now, increasing the rate of transpiration is very important. You understand this. Why would it increase the rate of transpiration? Because you see, when light intensity increases, photosynthesis increases and stomata open. This is in response to light, stomata open in response to light. So, when the stomata open, that results in more the stomata opening, more the rate of transpiration, because now the stomata open, so water vapors are going to diffuse out. Please don't think that light is giving it heat, because we would have a heat shield here. We would not allow the heat to pass, we'd only allow the light to pass. So what you have to understand is that increasing the light intensity means more the stomata open, photosynthesis is more. So more the rate of transpiration, because the stomata are going to allow the water vapors to diffuse out. If you compare this with the same potometer in dark, that would be a fair comparison. The rate of transpiration would be very minimal, because in dark the stomata close. So the reason why in light and more intensity is increasing, the more the rate of photosynthesis, more the stomata open, more the rate of transpiration. Don't think that increasing light intensity will increase the temperature. No, 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 no. It's not going to increase the temperature. Please be very clear about this. Now, look at this graph and let's see how you can understand the effect of humidity on the rate of transpiration. Whenever there's a graph, I always tell you a very simple mechanism is that you write 1, 2, 3, 4 on it. So just write 1, 2, 3, 4. And rate of transpiration, write anything 5, 10, 15, 20. So you see, when the humidity is 1, the rate of transpiration is somewhere here. When the humidity is 2, the rate of transpiration has decreased. When the humidity is 3, the rate of transpiration is lesser. So why is increase in humidity causing a decrease in the rate of transpiration? There's a very simple reason. You see, the water inside has become water vapor. Now this water vapor is diffusing out. But why would it diffuse out? It has to be more here and less here. Only then there's going to be diffusion. But imagine if it was the reverse. If it was 10 inside and 100 outside, well then I'm sorry, it's not going to diffuse out. It's not going to diffuse out. So if it is more humid, humidity means the water vapor content of the air outside the leaf. The water vapor content of the air outside the leaf. So as you can see here, if outside the leaf there is more water vapors and inside there is the leaf there's less water vapors, then why would it diffuse out? So more the humidity outside, lesser. This is what was happening to the rate of transpiration. It is decreasing. I'll just clean this graph so that you all can understand this. So as the humidity is increasing, the rate of transpiration is decreasing. The graph shows you this. This graph shows you this very clearly. The humidity is increasing and the rate of transpiration is decreasing. So more the humidity, the explanation of course is very simple. More the water vapors outside the leaf, lesser the chances of it diffusing out. The water vapors diffusing out. Well, the water vapor diffusing out is the rate of transpiration, the number of water vapors diffusing out of the leaf. First topic to be discussed is translocation. And it says describe translocation as the movement of sucrose and amino acids in the phloem from source to sink. Now from source to sink means source is the place where it is produced. So leaves would be the source. And sink would be the roots where no photosynthesis take place and they need it. So the sink is the place where they are used which is unable to photosynthesize and make its own food. Source is the place where photosynthesis take place or it is being provided. So source is the provider and sink is the user. So this is the important thing that you need to understand. That is why phloem movement is up and down, because if there's a leaf and there's a flower up, so from the leaf it's going to go up the stem and into the flower. But from the same leaf, the sucrose can also go into the roots. So that is why it is in two directions. Movement of sucrose and aminos in the phloem can be upwards or can be downwards. Now coming to this translocation process. Number one, sugar created in the leaves by photosynthesis. This is the source. So the source enters the phloem by active transport. So source will be the provider. Then when the sugar concentration in the phloem increases, water is drawn into phloem cells from the xylem by osmosis. Of course, this will create a high pressure. Number three, the addition of water from the xylem causes pressure to build up inside the phloem and pushes the sugar down. So the sugar moves down. Number four, sugar from the phloem enters the root cells, which is the sink. The sink is the user. So provider and user by active transport. So basically translocation is the movement of sucrose and amino acids in the phloem from source to sink. Source can be a leaf, source can be any green part, sink could be any non-green part. Even the white part of a variegated leaf will be a sink. A white flower will be a sink. A red flower will be a sink. Now this is some extra information which I want to give you all for a simple reason that you look at this graph and it is temperature and rate of photosynthesis, right? And you see a graph like this. The graph is increasing and then it is decreasing. So this tells you that photosynthesis is an enzyme controlled reaction, because this is a typical graph of enzyme. Enzymes after a certain temperature will be denatured and the rate will decrease. But look at this graph, temperature and the rate of transpiration. Now, as the temperatures increase, the rate of transpiration has increased, but then it is becoming straight. The rate has become constant. And the reason why, now look at this graph, this is for the very students who are very keen biologists. Increasing temperature has not affected the rate of transpiration. It's not a graph like this. It's not a graph like the one in photosynthesis. The reason why, because transpiration does not involve any enzymes. No enzymes are involved in transpiration. Transpiration is just evaporation and diffusion, so no enzymes are involved in transpiration. And then I've also given you the same graph, rate of transpiration and humidity. So you can see how I've now tried to develop a concept by which you try to understand is how why transpiration is not going to be affected by temperature because no enzymes are involved in it. Now this is a very profoundly difficult MCQ. Which environmental factors were varied to give lines X, Y, and Z? I'm not giving you the answer. I want you to pause the video here and have a look at it and decide which one is the answer. So you can see X as the environmental factor is increasing, X is decreasing. So that has to be humidity. Then Y you can say is increasing, environmental factor, temperature is increasing, so Y is temperature, and Z is the light intensity. So the answer is C. I know I do not agree with the Y part because of course they've shown such an environmental factor in which because temperature it should level out as well. But then because we have no other choice of that, so Z is light intensity and Y is temperature. So the answer is C. Please think about it, contemplate, and then check with the answer. I'm rubbing off the answer so that you all can then figure it out. So have a look at it, understand it, and see why the answer is C because the others don't make any sense. This ends this chapter. Thank you for watching. Please do subscribe. And if there is anything which you have not understood or is not very clear, please do leave in the comments and I will try to get back to you as soon as I can. Thank you very much and all the very best.